I would like to thank the organizer for giving us this opportunity to share our work. So this is my disclosure. I'm going to cover three different topics. First, we will be sharing some of our work in using expression data to how we control, how do we define molecular signatures for inflammation control in psoriasis. And I'm going to share with you some of our studies that use the epigenetic or genetics approach to define some factors that associate with the inflammation control or drug response. And at the end, I'm going to just share with you about what implication that we're going to have, like when we do the inflammation control in psoriasis. Okay, so over the past decade, there are different studies that have already been using gene expression to profile the psoriatic skin. This is a study that we did over 6 years ago. We tried to look into gene expression profiles for individuals that receive anti-TNF treatment. So the baseline here, basically, those are all the individuals, like we have all the non-lesional, lesional skin that being profiled, and you actually see really good separation in baseline. But when the patients start receiving the anti-TNF treatment, you can actually start to see the lesional skin kind of marching towards the position of non-lesional skin, right? So there's still some heterogeneity, but you can actually see this is like a global scale change. And the other way that we could look into is like through the heat map here. And then you could actually see like all the genes are right in here, and then you actually see the big differences between non-lesional, lesional at the baseline, and then you can see the when the patient receive by week 12 the anti-TNF treatment, then kind of normalizing towards the non-lesional skin. So this is like a global scale that we have been looking into. But then some of the key things that we are more interested in is to see how do we define certain inflammatory programs, and how do we know quantify whether they are normalized or not. So to look into this, we look into this like XR study that we just recently published earlier this year. The key thing is to try to still profile the gene expression changes, but for the patients that receive either ixekizumab or the guselkumab treatment. So similar to the anti-TNF cohort, we also have patients that have non-lesional, lesional skin in the baseline, and also the subsequent week when the patient started to receive both treatments. Okay, so this time, okay, we are more interested in looking into certain cytokine responses for keratinocytes, and especially the IL-17, TNF of IL-36. So we actually integrate the XR study with an independent genomic studies that we conduct using RNA-seq to profile the genes that are being induced by different cytokines in keratinocytes, in primary keratinocytes. And you could see that, so these are the data integration that we have, but then the heat map here is actually showing the genes that got induced by different cytokines in here. How many of those are actually enriched among genes got normalized by the two treatment groups under different weeks, okay? You actually see that there's like downregulated of the cytokine signatures, okay, especially for IL-17, TNF, or 36 post-treatment, okay? And then, but especially for ixekizumab, we actually could start to see the normalization as early as week 1. So we are now defining certain cytokine signature changes. So, the other way that we look into is actually try to see how do we quantify it, okay, using the normal skin as a reference. So instead of integrating these cytokine signatures with XR study, now we are actually doing an independent genomic studies only on the control skin of normal individuals, and then we define an empirical distribution of how the cytokine signatures would behave in the normal skin, okay, using that as a reference now. So actually you see that this is normal skin distribution, and that we are now comparing against those baseline or week 1, week 4 samples from the XR studies to see how much normalization they will exhibit. So this just using IL-17 as an example. Okay, you actually see the y-axis now representing the normalization. Okay, and both treatment increase the percentage of normalization for both the guselkumab and the ixekizumab group. Okay, but then for the ICSI group, this actually we see that it started normalization as early as week 1, kind of concording with what I just shared, and then it's actually achieving around 70% of normalization when using the control skin, okay, as a reference, okay, by week 4. The other way, that we also look into is that the normalization of y-axis is actually correlated with the PASI improvement on the x-axis, and this is especially true in the ixekizumab group. Okay, you actually see like large correlation. So this using the control skin independent information to kind of as a surrogate, we could actually also have an indication of associating with the treatment response, like in terms of the PASI improvement. So the next topic that I'm going to cover now with the gene expressions that shows this kind of like quantification. Can we reveal certain molecular signatures that could be associated with like inflammation control for psoriasis? So this is a BC slide, but what we did was actually perform single nucleotide multiome data to profile simultaneously gene expression, genetic epigenetic profiles for longitudinal, lesional, and resolved skin of the same individuals. But the key thing that I want to share with you is that for genes or peaks that got dysregulated in the lesional skin, how many of those actually got normalized in the resolved skin from the same individual? Okay, and you should actually see that the key message is that, for example, in keratinocytes, a majority of those dysregulated genes in the lesional skin got normalized for the resolved skin. Okay, and that's like different variations for different cell types. But the key thing is that the epigenetic marks is actually much less normalized in the resolved skin. So the epigenetic changes that get normalized, okay, is more subtle than the different expressed genes. Because we know that psoriasis is a complex genetic condition, okay, so one of the key things that we are interested to know is that for epigenetic marks that got dysregulated in the lesional skin, how many of those are actually overlapping with the genetic signals? And then there's actually, for over 100 psoriasis loci, 70% of them are actually overlapping with any epigenetic signals that we identify in the data set. But the key thing is that many of those are actually dysregulated in the lesional skin. Okay, so this is like almost 2 times than expected. Okay, and another interesting fact is that many of those kind of differential peaks in the lesional skin actually have a higher accessibility in the lesional skin, as you can see in here. Many of those are like fibroblasts, T cells, myeloid cells, or like keratinocytes in here. So would this high overlapping with the epigenetic marks with GWAS signals could be one of the factors that we see a certain epigenetic sites being more difficult to normalize? Okay, so this is one of the questions that we want to answer because individuals carry different genotypes, okay, for different signals associated with psoriasis. So we look into this data set that from Dr. Alder's lab in Michigan. So they profile over 1,000 attack-seq data, and then we try to look into how many of those are actually overlapping with the allele-specific chromatin accessibility sites. Okay, so there's actually over 1,000 data set with like 75 heterozygous sites that we identify. Okay, and then many of those are actually overlapping with the GWAS signals, okay, but only in the context-specific manners, which means that they are CD4, CD8 that are activated CD4 activated cells. Okay, when comparing with resting T cells, okay, they're like almost 2 times more overlap. Okay, so indicating that's actually the GWAS signals that overlap with these epigenetic sites have this context-specific effect. Okay, so just an example to show you. So this is actually one particular locus. That's an EQTL for this interferon lambda receptors, and you actually see that in the resting site, there's actually no openness. Okay, and when this is like activated, when CD8 free CD28 activated cells, there's actually more openness in the epigenetic marks. And you actually see that for these activated conditions, the two alleles, okay, again, these alleles are associated with psoriasis, actually exhibit different chromatin accessibility. Okay, so this is just one of the examples that I show in this figure. Okay. All right, so just like one quick thing about what are the additional role the genetic could play. Okay, so this is a study that we did like a couple years ago, trying to identify genetic signals that could be associated with drug response. So this is an example to show that a locus KLK7 is actually associated with drug response for anti-TNF. Okay, and then this particular gene is also being upregulated in the lesional skin of psoriasis, and then when receiving the anti-TNF response, the gene starting to come down, normalize to the non-lesional level. So we try to lock down this KLK7 gene in keratinocytes, and actually could decrease the pro-inflammatory response to TNF when in the keratinocytes. So the idea is to show that the genetic signals that vary across different individuals, they exhibit context-specific manners, kind of controlling how an individual could be susceptible to a certain different degree of activation, and in turn to affect the gene targets that they're going to affect in the same signal. But at the same time, they actually have some impact on drug response, because they actually have the effect on controlling the inflammations for the genes that play roles in the psoriatic skin. So in the last part of my talk, I'm going to just give you some quick examples about, and also echo some of the messages the other speakers mentioned about how the controlling the skin inflammation could have implication beyond skin. So as you know, psoriasis has many comorbidities. This is just one study that we conduct a couple of years ago, trying to show different types of comorbidities using the insurance claim data. And obviously, PSA is one of them. And as we just discussed about, PSA occurs mostly after psoriasis, after skin disease, and then this is actually a figure that show that when we look into the PSA psoriasis signals, PSA genetic signals versus the cutaneous only psoriasis genetic signals, many of those actually overlap. So actually they're sharing different genetic signals that exhibit in the similar pathways. So, but in addition to PSA, there are actually the other psoriasis comorbidities actually also are being with actually complex genetic conditions like multiple sclerosis, type 2 diabetes. And in fact, when we use the genetic and as instrumental variables to actually mimic the psoriasis incidence, actually we find that there are different comorbidities. Actually, psoriasis actually has causal effects on that include type 2 diabetes and multiple sclerosis. And again, echoing some of the message from the previous speaker, when we control psoriasis, actually this kind of disease that we found psoriasis have a causal effect on, we actually reduce the incidence once the psoriasis inflammation is being controlled. So with that, I want to conclude. We have 3 key messages. As I mentioned in the first part of my talk, with the gene expression, we were just using bulk genomics data, as an illustration. But we actually, to measure, we can actually tease out certain inflammatory programs that are actually being amplified in the lesional skin, and how we could quantify the normalization when using statistical or computational approach. And then there are genetic or epigenetic factors that will explain why some of the inflammation is more difficult, at least indirectly, to be controlled. Also, they exhibit, their effect in a context-specific manner. And lastly, there's genetic share list or correlation between psoriasis and its comorbidities. So contributing, that's why this could play roles in why when we control psoriasis, okay, some of the other comorbidities could also have the incidence rate being reduced. And with that, I would like to thank all my colleagues, my collaborators, and the members of my lab. Thank you. Happy to take any question.
Okay, we have time for a couple of questions. Anybody? I have, well, okay. Okay, go ahead.
Thank you. This was really fascinating. I have a quick question about the epigenetic changes that you've shown before, especially with the allele-specific changes that you see. Can you take that upstream and see if there are any specific epigenetic regulators that might be involved through, bioinformatics analysis of motifs or anything like?
Yeah, thanks. Great question. I didn't have a chance to go look into this detail. So remember, that's was one figure that I shared that showed how the epigenetic marks got dysregulated in lesional skin and how many of those got normalized in the resolved skin, right? So if you're only looking into those epigenetic marks that got normalized in the resolved skin, they're actually highly enriched with different transcriptional factors, including like AP1. So, but then obviously the next question is what are the upstream regulators for those epigenetic marks that are not being normalized? That's actually not enriched. So it could be all over the place, or maybe we do not yet have power. But yeah, this is actually a really good question. And then, yeah, I think AP1 is one of the key things that we show up, especially in T cells. But for the one that do not get normalized, we still need to look into that. Yeah.
Okay. Yeah. I got one question for you, Alex. So many of these kind of epigenetic modifications, they're through different types of mechanism. Part of it is methylation, some of it is histone modification, and it's pretty clear that as you kind of get the disease becomes kind of chronic, you're getting more and more of those epigenetic changes and kind of marks in the tissue. From those processes that are kind of involved, how can we use these tools and technologies that we have to really kind of map out the time frame where these epigenetic changes are kind of occurring, and how can we potentially reverse them?
Right. I think so. The animal model or the in vitro model would be key, but then for lateral cause, I think it's really difficult, right? Because so we have the genetic parts, that's the intrinsic effect, and then throughout some time there will be some activation that could induce some context-specific regulation, right? And then maybe the patient got treated, so some of them, but not all of them, got normalized. So then there's already diverse track, and then yeah, you have to model this. So yeah, I think in vitro or animal model could be first key to things. Look into it, but yeah, that's a
great. One final question, Li Hong.
Yeah, thank you. Thank you for your excellent talk. From my personal experience, because our center we're doing some bulk RNA sequence on the execution treatment before and after. But when I'm doing the KEGG and GL analysis, I was finding there's some type 2 diabetes, type 1 diabetes, the comorbidities pathway showing up. So I didn't know if you have the same experience with your data. Is this have a significance, indicating the correlation with the comorbidities with psoriasis? And my second question is that if you think about, they say that the obesity patient, they also have some skin inflammation. So have you ever think about comparing these psoriasis patient skin lesion with obesity patient without psoriasis lesion skin? Yeah.
Yeah, thank you for the question. So yeah, so besides type 2 diabetes, so I do genetics, okay? So besides type 2 diabetes, indeed BMI, okay, obesity has also been shown to have shared genetic signals with psoriasis. I mean, they're metabolic signaling. They're also immune signaling. I think those are the immune signaling parts that got sharing with psoriasis. When you do the animal model, for example, okay, there should be certain pathways that got induced, okay? I think that if those are the ones that got normalized, in psoriasis, for example, then maybe the other comorbidities would get normalized. But they're more complex than only one or two signaling pathway. So depending on how you induce, that, right? So yeah, so I think both ways. So sorry, I tend to give ambiguous answer, but this is, I think there's a lot of heterogeneity that we have to account for. Yeah.